give a little round of applause for her.
Hi, thank you everyone. Thank you for coming out this morning. I shouldn't have scheduled this on a Monday. Next time I won't. I am here to talk to you about surprising symbiosis, nature's unexpected partnerships. Okay, so first, you may be wondering who I am. I'm Bridget. I am, like Maria said, the Genomics Scientist at the Denver Museum of Nature and Science. I'm still relatively new, so I started in March of 2024. And basically what being a genomics scientist means is that I study anything that has to do with DNA. I grew up in Orlando, Florida, and I went to the University of Florida for undergrad, and then I went to CU Boulder for my PhD, and then I stayed on as a postdoc there. And also importantly for this lecture, I'm the curator and scientist on the new temporary exhibit called Brick Planet. Has anyone been to see Brick Planet yet?
Wonderful.
Yay! Okay, so I believe that you can go see Brick Planet after this lecture if you are so inspired. I'm biased, but I think that it's awesome, and everyone else seems to think that it's great too. So you should definitely go check it out. Hopefully this lecture gives you a little more scientific insight to that exhibit too. Something that you'll notice in Brick Planet and in this lecture is that I have a heavy bias towards microbes. It's my specialty. I'm a microbiologist. What are microbes? Microbes are tiny living things that live on and around humans, animals, and the environment. Microbes, like all living things, have DNA that can be studied. And if you hear the word microbiome, that is a community of many microbes living together. So I, as a researcher, am interested in microbiomes in all different kinds of hosts. So things like humans, their pets, and other animals. I should have silenced Teams, my bad. Okay. And I work at the Genomics Lab here at the Denver Museum of Nature and Science. Another raise of hands, who knew that we had a Genomics Lab? See Tiffany raising her hand in the back, which is good because she's the lab manager. The Genomics Lab is one of 3 public-facing labs here at the museum. The other 2 are all about dinosaurs. We're the only one that's not dinosaur-focused, which is cool. So if you ever go to the Genomics Lab, it is in the back of the Expedition Health exhibit. So if you go all the way through Expedition Health, all the way to the end, you'll find the follow your heart kind of maze activity. And then if you stop focusing on the maze, which is very fun, I know it's hard to do, and you look to your right, you'll see the Genomics Lab. There's a glass wall, and you might see myself working in there, or Tiffany working in there, or one of our volunteers, or another curator, or something like that. Feel free to wave at us. Nobody ever does. I feel like they don't know that we're there. So we always like to highlight it. The Genomics Lab, it is a functioning lab. One of the big questions that we get asked is, are we actors? Are we just putting on lab coats and pretending to do science? Absolutely not. We would never do that. We offer real lab experience for a lot of people. If you've ever heard about the Teen Science Scholars internship program, we support students through that metric. So we bring in high school students and we train them on how to do all kinds of wet lab work. We also accept volunteers. And we perform all steps of DNA sequencing at the Genomics Lab. So anything that we can do in-house, we try to do. And that includes microbiome research, disease screening, we dissect animals, we ID museum specimens, and we even do bioinformatics, so dry lab or computer work. This is all real research. So anything that we publish is going to be peer-reviewed research. We're supported by federal grants. We're also supported by the North Star Gift Fund. And we have complete oversight by ethics boards for anything that needs an ethics board, like human and animal research. And we also participate in the museum's brand new institutional review board. So we have our own ethics board right here. And of course, because we are public facing, there's a giant glass wall outside our lab, we have a focus on public outreach as well. Okay, so I've been using this word a lot, genomics. You might not know what that means. I'll explain it now. Every living thing has DNA. You have DNA, plants have DNA, dinosaurs have DNA, bacteria has DNA. DNA contains genes, which instruct organisms how to grow, how to look, and how to work. And genomics is the study of all DNA. So that includes genes and includes all of the DNA that isn't genes. How genes interact with each other and with the rest of their DNA and other living things, that's all parts of genomics. So we do a lot of genomic science here at DMS. It's not just me and Tiffany. So Tiffany studies how and Tiffany Nestle is our genomics lab manager. She was on the last slide. Tiffany studies beetles, and do beetle genes and microbes vary between different museums? I'll talk more about that project later. This is Garth. Maybe you'll recognize some of these people. Garth is our curator of ornithology, curator of birds, who studies how widespread malaria is in Colorado birds. This is Dr. Paula Cushing. She's the curator of arachnology, and she studies if spiders and ants that live together co-evolved. Of course, that's me. And I study how microbes keep us and other animals healthy and happy. So you can see that there's a lot of diversity, even in the things that we can do in the lab. And that's because we're all kind of at our core studying biodiversity. So when we think of biodiversity, we think of like everything living on Earth, which is a lot of different things. A lot of times we think about these really charismatic things like a bald eagle, the polar bears, the lions. It includes not only these big charismatic organisms, but the smaller ones, insects, arachnids, little tiny hummingbirds, plants. But an important part of biodiversity is the interactions between different species. And I'm going to bring up Brick Planet a lot in this because this is kind of where this talk originated from, was thinking about how in Brick Planet, which is the Lego exhibit, there's a lot of biodiversity represented. And when we think about trying to add science to an exhibit like that, it can be kind of daunting because you have to think of an aspect of science that can impact anything. All of these different things are represented in the sculptures in Brick Planet. So for the science, we wanted to focus on the interactions between these organisms, which is also known as symbiosis. So what is symbiosis? Basically, it's a close and often long-term interaction between two different organisms. We often hear the word symbiotic, and it kind of carries a positive connotation. We think, or we assume that it's a positive, good relationship, but that's not always the case. We are focusing on 3 different kinds of symbiosis, both in this lecture and in the exhibit. The first is mutualism. Mutualism is a relationship where both organisms in that relationship benefit. Commensalism is where one organism benefits and the other one is just kind of neutral, they don't care. And then the last one is parasitism, where one benefits and the other one is harmed. Okay, here's the other thing about my lectures, is I like to have them be interactive. So we have some trivia for you all. I'm gonna read out the question and then you just feel free and shout out the answer and we'll see how well you do on some of these questions. So, which of the following is not an example of symbiosis? We have A, a clownfish living in the tentacles of a sea anemone for protection while the clownfish provides food for the anemone. B, bees pollinating flower while feeding on their nectar. C, a remora fish hitching a ride on a shark and feeding on its leftover scraps. And then D, a hawk hunting and eating a rodent. What do we think? Which one's not symbiosis? D. So smart. Yes. D is not an example of symbiosis. This is just simply predation, but it's often confused for symbiosis, so I always want to clarify that. So let's go through these different kinds of symbiosis more in depth. The first one is called mutualism, and as I said, mutualistic relationships involve 2 organisms where both of them are benefiting from having that symbiotic relationship. So having this relationship often improves the survival, growth, or reproduction for both of the organisms involved. So for example, I gave the example of mutualism between bees and flowers. The bee is gaining nectar for food, and the flower gets pollinated, so that's helping the flower's reproduction. And there was a lot of examples already existing in Brick Planet. So actually in Brick Planet, just from the stepping back from the exhibit design point of view, we have no say in any of the sculptures that come in. It was all designed by the artist Sean Kenny. But Sean had kind of an emphasis on symbiosis, maybe if he didn't even realize it. So there's a lot of examples of mutualistic relationships represented in different sculptures. So the first one that you might encounter is the Galapagos tortoise and the finch. This is mutualistic, because this little finch comes in, it flies kind of around the head of the tortoise, and the tortoise recognizes this signal, and then they kind of stretch out their neck and their limbs and everything, and the finch comes in and flies around and eats parasites and insects off of the tortoise. So the tortoise gets a clean shell, and then the finch gets a really easy, stable snack. Another example, of course, we talked about the pansy and the bee. This one's right at the end of the exhibit. Another pollinator example is monarchs and milkweed. So sometimes they have a very specific symbiotic relationship with each other, pollinators and flowers. In this case, monarchs really survive because of milkweed. As caterpillars, they're eating milkweed, and that makes them kind of yucky to predators, so that's a survival mechanism. And then when they reach adulthood, of course, they're pollinating the milkweed and helping it reproduce. And then the last example I'll talk about in Brick Planet is there's a wonderful sculpture of the zebra and the wildebeest. They have a mutualistic relationships because of their participation in something called the Great Migration. You can read more about this in Brick Planet, but they travel really well together. They are both herbivores, but they eat kind of complementary foods. So the zebra eats the tougher, shorter grass, and that makes way for the longer, softer grass that the wildebeest prefers. And then they also help defend each other. So they have complementary senses. So one is better at hearing and smelling, and then the other one is better at smelling. So together they have kind of a team-building relationship where they are always on the lookout for predators. So that helps them both survive. Okay, I said I have a bias towards microbes. We're going to continue on the microbiome train. So here's a little piece of trivia. Which of the following can be part of a microbiome? Is it A, bacteria, B, viruses, C, fungi, or D, all of the above? What do we think? I heard A and D. The correct answer is D. So if you said A, you weren't technically wrong. But all of these things make up the microbiome, as well as other things. If you've ever heard of archaea or protists, those also can be part of the microbiome. So it's really a mix of anything that's microbial, anything that's too small to be seen with the naked eye. And there's a lot of microbial mutualisms in nature that I think are pretty cool. So many organisms have mutualistic relationships with microbes, so those organisms that are really teeny tiny. One of my favorites is the Hawaiian bobtail squid. These are really cute, first of all. They're so cute. You can't really tell from this picture, but they're very small. They would fit in the palm of your hand. Of course, they live in Hawaii, and because they're so small, they are really easy prey, but they're also predators. So they're not herbivorous, they eat other little sea creatures. They have a relationship with this bacteria called Vibrio fischeri, and something that you're seeing in this picture here is that Vibrio fischeri glows in the dark, which is really cool. So you might see from this picture how this squid has kind of a bioluminescent glow to it, and that's because of its relationship with Vibrio. So when the squids are born, they're born sterile, so they don't have any microbes living in their bodies. They quickly accumulate these Vibrio, and these Vibrio allow them to glow in the dark. And if you're thinking, why would you want to glow in the dark if you're prey? It's because it kind of works as a camouflage. So these are nocturnal hunters, so they hunt at night. So the way that they glow, if you look at this, it kind of looks like stars in the sky. So if you're looking up above at the squid, you can't tell that there's a squid there. You just look up and you see stars, basically. You think you're looking at the night sky. It's the same thing looking down at them, so it really helps them blend in the water column. So this helps them not get eaten and also not get spotted by their prey. And if they don't accumulate these bacteria, they will never glow. So they do some experiments where they purposely keep out these bacteria, and these squids can never glow without them. So I think that's a really cool relationship. You should look it up on YouTube if you get a chance. They're really cute. Another one is between termites and protists. So we know what termites eat, right? They eat wood. Not a lot of things can eat wood. I can't eat wood, you can't eat wood, I think. termites are able to eat wood because they have gut bacteria that allows them to do so. So they eat the wood up and they chew it really, really small, and then it moves into their hindgut where they have this really unique microbiome of bacteria and protists and archaea, and they're all called cellulolytic microbes, and that just means that they're able to break down cellulose. So the gut bacteria are getting a really nice, safe place to live in both of these cases. That's kind of the benefit for the microbes. And then for the termites, they're getting this really accessible food source that they wouldn't normally be able to eat. And then the last one is a little gross. It's koalas. We love koalas. Did you know that they eat their own poop, at least when they're born? So this adorable little Joey koala, when it's born, the first food source is its own mother's poop. And that's because, does anyone know what koalas eat? Eucalyptus. Eucalyptus is super toxic. It's nothing else is eating eucalyptus. Koalas are able to eat eucalyptus because they have specialized gut bacteria that help break down the toxins, and so it's non-toxic to the koalas. But they're not born with this specialized gut bacteria. It gets passed down vertically, meaning between generations, from mother to offspring. So the next time you look at koalas, think about them eating their own mother's poop. I know that's what I think about. And then the last example I'll give is the human microbiome, the one that we're all probably most familiar with. So we have many relationships with our own microbes. We have many good relationships, we have some bad relationships. It's kind of complicated. So I'll go through all these different kinds of symbiosis and how they're represented in our own microbiome. So there's a lot of mutualistic relationships. I would say that that's the predominant relationship that we have with our microbes, is that we're both benefiting. So our microbes, they get this really nice place to live. It's safe, there's constant food sources, it's awesome for them. And depending on where our microbiomes are, we get different services from our microbiome. So the big one that people think about is the gut microbiome. The gut microbiome helps us to digest food, helps us to harvest nutrients, and this starts at birth with bifidobacterium. So bifidobacterium is one of the first microbes that gets introduced to babies, and it helps us to digest things like breast milk. And development in humans from birth to around the first 1,000 days of life, that's around 2 years old, is key for gaining microbes that aid in immune system development. So having a healthy microbiome, it's been proven, can help with allergies, with building your immune system, preventing things like asthma and things like that. There's a lot of great developmental aid that we get from our microbiome. A lot of our microbes, though, are commensal, so they're just kind of there. They're not doing anything super good or super bad. A lot of microbes, you could say maybe this is mutualistic. They benefit us just by simply being there and crowding out things that might be harmful, like pathogens. So a lot of your skin microbiome, for example, is commensal. We don't really notice it, it's not really helping or hurting us. And then the last one that I think people fixate a lot is the parasitic kind of nature of some microbes. So if you think about E. coli, that's a big one. And I think that's really unfair to E. coli, justice for E. coli. Because E. coli is really mostly beneficial, and we like to say that microbes are only harmful when there are too many in the right place or too many in the wrong place. So it's really about finding that balance. E. coli, it is beneficial. It's supposed to be in your gut. It helps you produce vitamins. And it's just when it gets in the wrong place, and it happens to all of us. But anyway, that's a little overview about the human microbiome and our kind of symbiotic relationship to our own microbes. Okay, more trivia. So, what do you think is the ratio of human cells to microbial cells in the human body? So is it A, there's 1 human cell to 10 microbial cells, B, 1 to 1.3, C, 1 to 2, or D, 1 to 100? What do you think? D. Oh, I tricked you. Yeah, this number is way lower than you think it is. I mean, they're still outnumbering us. We're still mostly not human, which is interesting to think about. The number we used to project used to be closer to A. We thought that there was a lot more microbes relatively in our bodies, but now they're pretty much equal and they have a slight advantage. So sometimes I like to think about that. But who's really driving the ship? I don't know, not me. Okay, so let's move on to the next kind of symbiosis. So this is commensalism. Commensalism is where one organism benefits and the other one is really neither helped nor harmed. This one can be really tricky. This is the one where I present things and then people are like, but what about this? And I'm like, you're right. It comes with a lot of different opinions. And it's one that maybe you first describe a relationship as commensal, and then you learn more information and it transforms into something else. But one of the ones that people mostly agree on, one example is barnacles in whales. So barnacles attach themselves to whale skin. The barnacles gain mobility, they get access to nutrient-rich waters just by catching a ride on the whale, and the whale doesn't experience really harm or benefit from their presence. There's a couple examples of commensalism in Bird Planet that we highlight. So one is, there's this, I actually really like this sculpture, I'm just gonna talk about this. I think it's really pretty. I think the ocean part of Bird Planet is really stunning. So if you go, make sure that you look at it and check out the lighting, because I think the lighting's really good. And our exhibits team did a great job on that. Seahorses and coral have a commensal relationship. You can see it in the sculpture, how this seahorse is looping its tail around a branch of the coral. And that's just for stability. So the seahorse, they're floating around in the ocean. Sometimes they just need a place to rest. So they latch onto the coral. Of course, as water flows by, they're able to get food. It doesn't really hurt the coral. They don't really care that the seahorse is there. So the seahorse benefits, the coral is just neutral. Another example that we talk about in the exhibit is sea turtles and fish. So sea turtles, the way that they feed is at the ocean floor. So they kind of stir up things at the ocean floor. By doing that, they're creating a food source for other fish to come through after them, and then they get easier access to food that might have been buried in sand. So in that example, the sea turtle is neutral, the fish are benefiting. Okay, some other cool commensalisms just in nature. Again, all of these, you could argue with me, you're right. Hyenas and lions. Typically a lot of scavenger relationships, maybe somebody's getting annoying to the other one, but they can kind of be thought of as commensal. So if something is coming in, like a big predator like a lion, they're killing something, they're eating what they want, and then they're leaving a carcass, the hyenas come in after and they get basically food without having to work for it. So in this case, the hyena benefits, the lion not so much. Another one that's similar to the seahorse and the coral is shrimps and sea urchins. So sea urchins, obviously they have these very pointy, very sharp venomous spines, which is a great defense mechanism for the sea urchin. It's also a great defense mechanism if you're living inside the sea urchin like shrimp do. So shrimp are teeny tiny, they kind of live within the spines, and because of the natural defenses of the sea urchin, then they get the benefit of also being protected. The sea urchins don't really care though. The last one, this is my dog, by the way. Ancient dogs and people. So right now, you can think of dogs as having a very mutualistic relationship with us. I know that I have a very mutualistic relationship with my dog, where she brings me joy, and I do literally everything else for her. And it didn't always start this way. So it kind of started more similarly to the hyena and the lion, where again, people, we were hunter-gatherers, we were kind of doing our own thing. Dogs would follow us around, they'd get really easy food sources, and then that commensal relationship turned into more of a mutualistic relationship, or parasitic, depending on how you feel about your dog. So that's an example of how some relationships can change. Like scientists might describe something as commensal, and then they might change their mind, and it might become something else. Okay, so more trivia. Which of the following examples does not exhibit commensalism? And I picked all plant examples because I feel like we've been ignoring plants a little bit, but they all have symbiosis too. So is it A, an orchid on a tree trunk, B, mistletoe plants on tree branches, or C, Spanish moss on an oak tree? Which one is not commensalism? B, C, it's B. The this is very apropos for Christmas time. Mistletoe, we love it, but it's a parasite actually. So this is an example of parasitism. With these other plants, like an orchid or like with Spanish moss, this is commensalism because these plants are getting stability from another tree or something. So they're getting structure, they're getting physical support, but they're not taking away any resources from the tree. That's not the case with mistletoe. Mistletoe latches onto a tree and it redirects nutrients like water and nutrients to itself. So it's actually hurting the tree, but it gets a lot of benefit. So that leads me into the last kind of symbiosis, which is parasitism. So parasitism is that relationship between two species where one organism, the parasite, benefits at the expense of the other, which is the host. Parasites often need their host to survive for a prolonged period to continue obtaining resources. It doesn't really do parasites any good if they kill off their host right away. That would just be an example of predation. So for instance, this lovely example here is a tapeworm. A tapeworm can live inside a host intestine for years without killing the host, although of course it might cause other issues like malnutrition and other health issues. In Brick Planet, there's not really very called out examples of parasitism. We do talk about one which you're like, there's probably a tick in here somewhere. There's a really great deer family. There's a buck and a fawn and a doe. So deers and deer ticks, that's a really common one that we hear about all the time. In this case, the deer is the host, the tick is the parasite, feeding off of the blood of the host, which can cause illness, it can cause sickness in the host, but the tick is having a great time. Okay, some other, I hesitate to call them cool because they're not that fun, but here's some interesting parasitic relationships in nature. So one of them that I think is really interesting and one that I've studied before is this relationship between mud shrimp and isopods. So mud shrimp are, you'll see it in the next slide, there's a nice video of them, but they live in mudflats all along the west coast of the US. And they have a parasite called Orthionegrophensis, and that is a little isopod, it's a parasite. And the shrimp is called Eucogebia, that's the name of the genus. They're really important, they're ecosystem engineers. They live in these mudflats, and they build these kind of Y-shaped burrows here. So they transform the environment around them. And you can see from this diagram, there's lots of other little things that also live in these burrows. So a lot of things are also benefiting from the shrimp being here. And the shrimp depend on their microbial symbionts, their microbiome, for their health. Something to note about parasitism is that it can disrupt the other forms of symbiosis, so it can disrupt mutualism and commensalism. Orthione is an invasive species. It came over from East Asia on ballast water, not good. It came over to the US West Coast, and it has infected all known populations of native mud shrimp, which is Eucogebia pugetensis. So I think that's kind of a crazy fact. Every single population we found has been parasitized by this isopod. There's also other non-native shrimp that live here, and they have developed some kind of resistance to this isopod. We don't really know why. That's an ongoing research question that is actually being studied right now at CU Boulder. And the hypothesis is that there's dysbiosis in the microbiome of the shrimp that makes the host more vulnerable to the isopod coming in. So you'll see in this next, I put in a little video so you can really see the shrimp in the isopod. The isopod kind of tucks under the gill flap of the shrimp, and it sucks on the hemolymph, so it sucks on the blood of the shrimp. It takes away all of the resources for the shrimp, and then they are effectively castrated, so they can't reproduce anymore. So I don't know that there's been like a consensus on the endangerment status of the shrimp, but they're certainly in decline because of this non-native isopod. And because they're in decline, they're not building these burrows anymore. These other things, like there's different bivalves, there's mollusks that also live in these burrows, so a lot of things get affected when parasitism comes into play, so it disrupts a lot. And I'll show you, okay, before I play this, you might have heard of this shrimp isopod relationship before. For some reason, in 2020, it really took off on TikTok. I don't know why, but this is one of like so many different TikTok videos, and I'll just play it for you. I would love to hear if you know why it went viral, but I don't know.
This little shrimpie's name is Nancy, and her body's being infested by parasites, like this parasitic clam on her abdomen. Get out of here, that's where Nancy's eggs go. Nancy also has a bulging, pulsating invasive parasite under her gill flap. This parasite's called Orthion griffinus. It's an isopod parasite that's native to Eastern Asia, but found its way over to America, destroying the shrimp's populations. We're gonna get this parasite removed so that Nancy can live a happy, healthy life and have babies one day. But this parasite's so big and chunky, it's so hard to get out. Anyways, I removed the parasite, and take a look. We'll even remove the small male parasite that latches onto the giant female. Yo, nature's flipping weird. Next up is Rita, and she just molted. You can see the parasites through her gill flap. Since she just molted, her body is soft as jelly, so we gotta be extra careful. Notice how the parasite's legs hang on for dear life. I removed the parasite, yeeted them to my good friend Franklin the sea chicken, set my shrimpy babies free, and be sure to follow if you love ocean animals.
Yeah, I think it's just really satisfying seeing him like peel off the shrimp, the isopod from the shrimp. I did some field work with that project at CU Boulder, and I went into the field. I went to Oregon, I think, and it is satisfying. I have to say, it does feel good to take off the isopod from the shrimp. Okay, so let's go to one more cool-ish parasitism, and this is another one that you might have heard of. Cats, toxoplasma, and us.
Cats are cunning, stealthy hunters. And for prey like rodents, the smell of a feline means stay away. But this rat is heading straight toward danger. It's been tricked by a microbe. The story begins with a cat infected by a microbe called Toxoplasma gondii. The cat's poop teems with this parasite's eggs, and rat will eat almost anything. Toxo invades the rodent's brain and reduces its aversion to the cat's scent. Now acting boldly, the rodent is easier for the cat to catch. Toxo only reproduces in a cat's gut, so when a cat eats an infected rodent, the cycle begins again.
Okay. Ew. This is why just be very careful changing your cat's litter box, because that's how toxoplasmosis can transfer from the cat to a person. Okay. More trivia. I think this is the last one. Okay, which of the following do you think is not an example of parasitism? This is easy. Is a cat and toxoplasma. Vampire bats and large animals is B. Or C, bark beetles in Colorado forests. Which one is not parasitism, do you think? B and C, I'm hearing. The answer is C. I didn't know this until we started working on Bird Planet, actually. I had kind of assumed that this was a parasitic relationship, but this is an example of how a relationship that is just kind of predation or can just be neutral can become really bad or exacerbated by human intervention. So bark beetles is, if we live in Colorado, we hear about bark beetles all the time, how they are destroying forests and they are so bad, and cut the beetles a break, like it's not really their fault. The forests are weakened because of things that are out of the beetles' control. So we are preventing fires, it's drier because of climate change. There's all of these human-induced factors that are weakening forests and just allowing really this predatory relationship, this herbivorous relationship, to get kind of out of control. Vampire bats and large animals, that is an example of parasitism. So not only bats, but there's also vampire finches come in and they feed on the blood of larger animals. So in that case, the vampire whatever bat finch is getting a benefit because they're getting blood as a food source. It does hurt the larger animals though. Okay, and then I think I have a little time left, so I'm just going to talk about some of the current research that's happening in the genomics lab that's related to symbiosis. Spoiler alert, it's all going to be microbial, but if you go through Brick Planet, there's a really nice panel at the end of the exhibit, and that talks about Garth's research with symbiosis, it talks about Paula's research with symbiosis, so you can also read that. I just don't want to speak for them. I think Paula has given a different talk too, and Garth gives talks all the time, so you should definitely come and hear about their research directly from them. Okay, so right now we have 4 projects kind of in development or in progress in the genomics lab. You may have heard of some of these before. We just wrapped up the green therapy study. We have a study called the Domestic DNA Discovery Project. We also studied dog gut microbes and behavior. And then we're just about to start this project, which is microbiome at the museum. So first, the green therapy project is a human subject study, which is at DMNS. Something fun about the genomics lab is that we can conduct human subject research. So if you were here for genetics of taste, all of those different studies, you should talk to Tiffany because I wasn't here for those. But that's another example of human subject research, which I think is really cool. It's something very unique to us as a natural history museum that we're able to do human research. So this means that we were enrolling real people just like yourselves, both staff and guests of the museum and volunteers. And we were investigating the connection between people's time spent in green spaces. By green spaces, I mean parks or gardens, anywhere where there's green plants. The human nasal microbiome, which is all of the bacteria, all the microbes living in your nose, along with associations between people's pets and their self-reported wellness. So we've already enrolled for this study. We enrolled 111 participants in summer of 2025. So we generated survey data from people providing their demographic information, behavioral patterns, like how much time they're spending with pets, with green spaces. We swabbed people's noses, so it was very non-invasive. It's very similar to if you do like an at-home COVID test. That's kind of how you can swab the human nasal microbiome. And then we're collecting green space data now. And data is being analyzed now, so you'll have to stay tuned for results. But I believe Tiffany and I are giving a lecture in May of next year. So we're hoping to present results for this project and the next one that I'm about to talk about. So the next one is called Domestic DNA Discovery. We know that domestids play a pivotal role in vertebrate prep labs in museums, including DMNS. So domestids are flesh-eating beetles. Did you know that we have flesh-eating beetles right here at the museum? Some of us. They are our only living collection of beetles. Of course, they're only eating decaying flesh. It would never come in and like eat you. They're very cute, I think, actually. They live in our vertebrate prep lab, so they're kind of little employees of the museum. When we get a new specimen in, like maybe something passes away at the zoo and then it's coming to us, there's a lot of processes that have to happen to make that specimen usable for us as a museum collection. One of them is skeletonization. So you're kind of removing the skin, you're removing most of the flesh from a specimen, and then you can put it into a colony of the flesh-eating beetles, and they skeletonize the specimen for you. So they eat off all of the flesh. They do a lot of the work for us. We just do some light cleaning of the bones later, and then they're ready to go in the collection. This is not unique to us as a museum. Pretty much any natural history museum that's accepting specimens that has a prep lab like this, they're going to use the same kinds of techniques, including these beetles. So we are investigating the genetics and the gut microbiomes of these flesh-eating domestic beetles across museums nationwide. So we've partnered with 8 different institutions. I guess we've technically partnered with 7. And we've collected beetles from all of these different museums across the country. We collected them in the spring and the summer of 2025. Something really interesting about these beetles is that they are really inbred. So you get a colony of beetles, and then they can just live in your museum and replicate and reproduce. you have this kind of infinite source of little worker beetles, but that makes them genetically, they kind of merge into one. We've seen it with our own beetles, and we're starting to see it with other institutions too. So like our beetles have been here for at least 40 years, around 40 years or more. At the American Museum in New York, they've had their beetles supposedly for the past 100 years. So we're really interested to see how genetically similar or different they become. And then beetles are also a great way to study the gut microbiome because you're controlling a lot of factors with them. They live in this very controlled environment. Genetically, they're pretty much clones of each other, and then you know exactly what they're eating. So we're also looking at maybe how they've adapted with different gut microbes depending on what they're eating. So again, data is being analyzed, and this is the other project that I believe Tiffany and I will be talking about in May of next year, if you're interested. We're also starting a collaborative project looking at dogs, dog gut microbes, and behavior. So we know that, at least in human subjects, there is a kind of a brain-gut connection in many people. The gut microbiome has been implicated in things like mental health, but does this apply to our furry friends? So we're collaborating with other researchers. We're collaborating with cohorts of shelter dogs and working dogs to investigate the potential connections between the gut microbiomes of dogs and then different metrics of their behavior. So if they are a working dog, how trainable are they? How quickly do they pick up the things that they need to learn? If they're shelter dogs, does this impact other aspects of their behavior, like aggression? So this one, we've just received a lovely shipment of dog poop, so that is waiting for me. That's a project for January, I think. And then this, I haven't talked about this one before, but I think it's interesting. So we know that microbes are all around us. There's microbes on you right now. There's microbes on these chairs. There's microbes on this mic. There's microbes on your phone. Anything around us, the air, dust, anything in our built environment also has a microbiome. I think that's really interesting. And we exist in this really unique institution where we have a lot of high-touch exhibits. So we want to study how the microbiome of a high-touch exhibit interactive changes over time from installation to after heavy use. I'm not going to tell you which one it is, but we're getting a new interactive, and we're just going to start seeing, like, how does this microbiome develop? It starts with nothing, and then does it become more like people? Does it become more like dust? Does it become more like the plants outside? We don't know yet, but we're gonna start looking at the built environment too. So those are a few of the projects that we're working on in the Genomics Lab. Of course, feel free to ask any further questions if you have any to me, or I'm gonna volunteer Tiffany because she's here too in the audience. But basically what I want you to take from this talk about symbiosis is that relationships are really complicated. We all need each other in some way, and to conserve biodiversity, we also have to protect the different relationships that species have with each other too. So we need to conserve relationships as well, even the itty-bitty ones like microbes. With that, I'm happy to take any questions, and thank you so much for listening and coming out this early morning.
All right, like I said at the beginning, I'll pass the mic around. You can raise your hand and Bridget can call on you. And then if you speak into the mic so everybody here can listen, and for the recording, everyone can hear your really awesome questions. So, all right, I'm gonna do it first.
Thank you for your presentation. It was fabulous. The question I have is, when you get into isolated communities, specifically the International Space Station, where you get people up there for 6 months at a time, have they noticed changes in the biomes of the astronauts to the extent that it may produce negative health effects? And how would this ultimately affect Mr. Musk's idea of colonizing Mars?
Great question.
That's a really great question. I should know the answer to that, and I'm pretty sure that it's been studied. You're talking about, I'm assuming, the microbiome of the astronauts, like the microbial communities. Of course, they're bringing whatever microbes they have with them. When you reach the age of about 2, your adult microbiome is pretty much formed. So you're pretty stable from the age of 2 on. Any of the big changes that you're getting in your microbiome is from some kind of outside influence. So your environment, if you think about, I'm going to take antibiotics, that's kind of like wiping out my microbiome. I am not familiar, I don't think they do any kind of like sterilization process or anything. I would guess that it's probably fairly similar. I'd have to look up the answer to that question though. I would guess that maybe you would start to merge, be more similar to the people that you're on the space station on the space station with. Like if you compared those people to people on Earth, you would maybe see a difference, or you'd see like statistically they're a little more similar. That's a great question. I'm gonna have to look into it, but that's just my educated guess. I think we should be on Earth. It's just my personal belief is that maybe we should put a little more resources into safeguarding Earth before we try to colonize other planets. So that's all I'll say about that.
A little of a follow-up question from me. Does space have a microbiome?
Space, like, as a concept, I don't know.
Okay.
It doesn't have a microbiome. There's evidence, right, like on places like Mars that it used to have. We think that there used to be microbial life on Mars. So it's definitely like, if you believe in aliens, which I do, I think that they're probably microbes. I think there's lots of microbial aliens probably on different planets, but like space as a concept is sterile, yeah.
Okay. All right. Here you go.
We know there are negatives for taking antibiotics in terms of the mutualism. Are there negatives for taking antifungals? Are fungi, yeast mutual, or parasites in the human?
So fungi and yeast and things like that, those I would say fall into the same kind of relationship as bacteria and archaea and things like that, where mostly I would say it's a mutualistic or commensal relationship. Antifungals, yeah, it'll kind of do the same thing where it'll wipe some out. There's certain fungi that we know are really beneficial. I'm trying to think of which ones they are. It starts with an S. But there's a lot of yeast that are really good for you. Of course, like it's the same thing as any other microbe, where if you get too much or you get it in the wrong place, it can turn into that negative relationship. But yeah, if you take an antifungal, if you take antibacterials, then you're kind of starting from zero. You do rebuild that microbiome, but your defenses can kind of be down for a while. So that's why I think, antibiotics are great, but you should only use them when you absolutely have to, because I've noticed it even in my own body. Like, you take antibiotics and then a whole slew of other things can come in, like things you're not even thinking about, because it just wipes out everything. So they're great when they're needed. Otherwise, I always warn people against, like, don't take antibiotics for the common cold. Common cold is a virus. It's not going to do anything. You're just going to hurt your body more. Yeah. Good question, though.
Did you have a question?
I'm interested in your laboratory. Can you briefly describe what kinds of instrumentation you have? Is it in this building? How many people work in the laboratory?
Yeah. So the Genomics lab is a party of 2, and we're all in this room right now. So it's me and Tiffany. And Tiffany's raising her hand just back there. We are the 2-person, we're called the Health Sciences Department, technically. So there's 2 full-time staff. Tiffany's the lab manager, I'm the Genomics Scientist. Any projects we conceptualize together, so we're very much peers, we're a team that collaborates. Right now, we have 3 or 4 volunteers technically. We kind of bring on volunteers. At this point for microbiome projects, we're really looking for volunteers who have lab experience before. But when we do something like the Teen Science Scholars Program, you don't need to know anything about lab work. We can come in and we can train you on how to do lab work, how to be sterile, how to pipette, how to do DNA extractions, PCR, things like that. In terms of instrumentation, the things that we can do in-house, we can do DNA extraction, and that's just literally taking DNA out of something. So you take a sample, you're kind of you're opening up the cells, removing the genetic content, and then purifying that. That's basically what DNA extraction is. Using the purified DNA, you can do something called PCR, which is called the polymerase chain reaction. So you can amplify, or you can kind of make many copies of that one part of the DNA that you're interested in. So maybe it's a specific gene. You can make many copies of the gene and then study that. We are able to do some sequencing, and that's just kind of getting the letters of DNA back as like a data file. Mostly we send out our materials to another institution for that part of the process. So one institution that we collaborate with is CU Anschutz, which is right down the road. So we work with them for a lot of sequencing. Other than that, we have microscopes, we have centrifuges, we have pipettes, we have anything that you would use for the first few steps, from dissection to DNA extraction to PCR to running a gel. We can do that in-house. Past that point, you're getting into very expensive equipment that's kind of hard to maintain for us with party of two. So we send that out. But yeah, like we're interested eventually in taking on interns or research assistants or something like that. We're just trying to get data back first and build up work for volunteers and interns to do. But great question, thanks for asking.
Thank you.
You can find her office on the second floor again in a glass cage, office. And you can peek and knock on the windows and get her attention too. Just don't spook her too quickly, all right? That's a second floor.
Hello, I have a few questions, but I think they're quick. The cat and the microbe, you said that was parasitic. What's the effect on the cat from the microbe?
Oh, that's a good question. People ask about that because a lot of the times there isn't really an effect on the cat. It's kind of the same with people, where if you're not healthy, there is an effect. So if you're immunocompromised, that's why they tell pregnant women not to change litter boxes. It's because you're a little bit more immunocompromised, and then you would get sick from the parasite. So the parasite changes behavior in the rat. If it gets into the cat, like it's using the cat as a means to an end to reproduce. But if you have a cat that's not 100% healthy, I think it can cause, just some kind of illness, sometimes some seizures or something, inflammation, liver disease, I think. It can cause a few things. And then in people as well. Mostly it's neutral, but if you're immunocompromised, it can have effects. So you can get like a fever, you can feel like you have the flu or something like that. So it's not across everybody, it's just in some.
And then you mentioned vampire finches. This is a bird that's a vampire?
Yeah. It's a finch, and it does the same thing where it has a very sharp, pointy beak. You should ask Garth about this because he'll know more about it, but it will fly in, and I think it's like livestock and things, or like big, large mammals. I'm not sure. It'll come in and kind of like poke at them and draw blood, and it will feed off of blood, is its food source, yeah.
Wow. And then my last question is about the beetles.
Yeah.
And because they've been isolated colonies for so long, when would we ever see them become different species that they couldn't interbreed?
Oh, that's a good question. We define species in a few different ways. Right now, they're definitely all the same species. If we were to sample all of your genomes, or even all of your genes, like if we took one specific gene from each of us, you would naturally see a little bit of variation with all of us. Like there's 4 letters of DNA, there's A, C, G, and T. You would see some people have an A here, some people have a C or a T or something like that. There's a bit of natural variation across all organisms. With the beetles, we're seeing that there isn't even that. Like they are literally letter for letter identical. We use kind of like a national database to match the genes and identify the species. That's what we do if we have like something come in, like a roadkill or something from a wildlife reef habitat, and we're not sure what the species is. We'll take DNA out of it, we'll match to the database, and then it'll give like a % match to DNA that exists in the database. And I'm forgetting the magic number, Tiffany probably knows, but beneath a certain percentage point of similarity, that's when you start getting into, okay, maybe we can call this another species. I think it's like 94%? About 94%. Yeah, so at that point, that's when you could start thinking about, okay, maybe we can call this a different species. But there's other things that go into play, especially with things that kind of like, I hate to call them macrofauna because they're really tiny. But with big things, you can look at the morphology, you can look at the physical structure, how they look, and then that can help you define different species too. So right now, they're definitely all the same species, but they're becoming very highly specific to the institution. They're becoming more inbred within each colony. But I think if you put in new beetles, they would reproduce fine. And we're even seeing that with some of the colonies. Like, we've partnered with the Bishop Museum, and that's in Honolulu. I think I'm saying it's the Bishop, right? Mm-hmm. Where some wild dermestids, we think genetically, have gotten it, and they're starting to reproduce with them. Because their colony, ours is like within many layers of wall, and it's very highly guarded. And theirs is a little less so. Like, theirs is outside, and it's very reasonable to think that there are some wild beetles getting in. And they seem to be doing fine, and it's probably good for the colony. So, yeah, I hope that answers your question.
Yeah, thank you.
Okay.
I'll sneak in just across. Do you mind passing this down?
This is about when you said newborn babies start with a certain kind of microbiome. So I have two questions about that. Where does that come from? And do all species have a similar progression of microbes?
That's a really good question. So when we think about where microbes come from, there's some debate with people. I'll tell you what I think, but don't be surprised if you hear something different from somebody else. Some people think that babies are born completely sterile. I don't think so. I don't think that you can really say that. It does depend on how babies are born. So if you were born vaginally, you are picking up microbes just from being born vaginally. If you're born via C-section, it's been proven that you have less microbes at the start. You're born more sterile than somebody who was born vaginally. So when you get microbes from your parent, that's called vertical transmission. So it's called vertical because you think of generations and it's being passed down from one generation to another. You can also acquire microbes horizontally. And so horizontally is like, I'm in this generation, I'm not getting it from any other generation. So that's things that are like environmentally acquired. So in the instance of human babies, a food source, whatever food source that they're having as an infant, that can also dramatically impact the microbiome. That's something I used to study at CU Boulder. If you look at babies who are being breastfed versus formula fed, there's definitely differences in the microbiome there. You combine that with how babies are born, vaginally and C-section. You compare that to lots of other environmental impacts, like are there pets in the home? Do you live in a big family? Do you have lots of siblings? Are there, do you live on a farm? Are your parents super clean and they like Clorox everything? All of those different things can impact the microbiome. So it's really hard to say. I would say mostly like bifidobacterium you're getting. I mean, even if you're breastfed, you're getting microbes that way. Bifidobacterium is one that you're probably getting from a parent, from your mom, yeah.
And do other species have a similar progression?
Yes. It depends on the species. A lot of people, again, will say every single living thing has a microbiome, and that's not necessarily true. We sometimes think about something called the core microbiome and then like a transient microbiome. So a core microbiome is just, you have this thing in you all of the time. It's very stable, it doesn't really change. A transient microbiome is the things that we're getting from our environment. The things that we're picking up that kind of pass through us, we're using them, but they're not sticking around in our bodies that long. So some things don't have a core microbiome. Caterpillars are one of them. They don't have a microbiome. For other species, it depends. It depends on the species. Something like the Hawaiian bobtail squid, that glow in the dark, that bioluminescent bacteria, they're not getting that vertically. They're not getting it from parents. They have to acquire it from seawater. So if you put them in sterile seawater, they're never going to have a microbiome. I used to study snails randomly. I studied snails, and snails are really easy to breed. And we could look at different generations of snails, and they would pass down microbes from parent to offspring. Similar with something like termites, a lot of those microbes that are very necessary to survive, to break down food, those will get passed down from parent to offspring. So I hate to say it, but it does depend. But there's a lot of evidence for both across the natural world.
We've got time for one or two more questions. And you've got one right here?
Thank you. I have two questions. First one is, what does the museum feed the flesh-eating beetles? And the second one is, after COVID, what did the studies show when people were inside more, using more sanitizers and their biome? Any studies there?
Okay. First question about the domestic beetles is what do we feed them? We feed them animals. We feed them anything that's coming into the vertebrate prep lab, which can be things as big as, like, when I started in March of 2024, there was a giraffe that had died at the zoo, and so there's a giraffe that lived in the prep lab for a long time, to even really small things, like if we get donations of birds or chipmunks or something from a wildlife rehab or something unfortunately didn't make it, they'll offer it to the museum, and something small like that might go into one of the smaller colonies. We actually have 8 colonies of the flesh-eating beetles at DM&S. So colonies 1 through 6 focus on the smaller animals. So those are kind of smaller, like terrarium size, like fish tank size tanks of beetles. And then we have 7 and 8, colonies 7 and 8. Those are more likely to house the zoo animals, so like that giraffe, or I think there's like, I don't know, you'd have to ask people who have worked here longer. There's been a lot of stuff that has passed.
We've had wolves. I think a month ago, a bird ran into the glass where you guys came in, and we didn't know what to do, so we asked our scientists, and they said, give it to the beetles.
Yeah.
Sometimes it's literally just cleanup for us, and then sometimes we're actually, like with the giraffe, have specimens that we're going to use scientifically or observe scientifically, and we want the science from those animals. So some of it's cleanup, some of it's actual science.
Yes, thank you, Maria. So there's a lot of stuff. They're definitely not going hungry. Something interesting about them is they have a growing cycle. So even though they're in this tank with like no natural sunlight, they can't tell it's the same temperature all year round. They kind of go into a lower level of metabolism around now, like through the winter. Even though they have no idea what's going on, they'll eat less. In the summer, they're eating like crazy, so that's really interesting. And then they also apparently have a lot of preferences. What's the thing that they really like? Reptiles? Is that it? Yeah, they really like reptiles. They don't like fishy things or blubbery things. Yeah.
Not a fan of seals.
Yeah, so I think they're, anyone who works with them are like, they're so cute, they have such personalities. If you ever get a chance to see them, you should say yes and just like plug your nose, because I think they're really special. And then your other question was about COVID and about how that affected people's microbiomes. I haven't studied that directly. I think, you know, it's one of those things where we have different relationships with different things. COVID is a virus, and so there's some debate about viruses. They're part of your microbiome too. We tend to think of them as bad, but there are a lot of good viruses that live inside of your body. So it's the same thing where they're getting a bad rap just because of a few pesky guys. But mostly they're supposed to be there. They're supposed to be good. Again, I'm going to have to look into the literature and see if I can find something about that. I would guess that maybe it's not as bad as I would assume that it is, because with COVID, I know a lot of people, including myself, we were going outside a lot more because you could be socially distanced and you could go for a hike or something like that, and you're picking up good microbes that way. Anyway, it probably did kind of shift our microbiomes. We probably became a lot similar to anyone in like our pod or like our household or things like that. But yeah, that's a really good question. Those are just my hypotheses. I'm not sure. I'm sure somebody studied it.
Someone fact check Bridget.
Yeah.
All right, we've got time for one last question, and then we'll wrap it up.
This is more a comment rather than a question. When we talk about the microbiome, most of us think about the germs that infect our guts. But I'd like to remind people that our skin is also covered with a microbiome. There are literally trillions of microbes on everybody's skin. And taking a shower every morning does not wash them away.
Yes, I love to think about my microbiome. I'm so happy everybody's along for the ride. There are lots of different microbiomes, you're absolutely right. Anywhere that your body interacts with the environment, from your gut, because you are consuming things, you're eating food, your skin is impacted by the environment. You have an oral, like a mouth microbiome, you have a nose microbiome, you have an armpit microbiome, and they're all very important. So don't be afraid of your microbes if you take anything away from this.
Thank you so much. Let's give a round of applause. Bridget will be around for about 5 minutes for any questions that we didn't get to, but just a reminder is that today is a free day, but for morning lectures, general admission is included in your ticket. So as always, you're welcome to exit into the museum. So that's as you exit, go to the left through Space Odyssey, or the way you came, which is going out the right. We appreciate your time, and we hope to see you in February next year. Thank you.